reactions. This would be a very good topic for the students, and students only a little number 9 have started, but sir, we started, sir.
Okay, no problem.
Okay, sir. Welcome you, sir. Please.
Good morning to all the participants who have joined till now, and expect the number to increase. First of all, I will introduce the basic concept of antigen, because in immunology, the two basic principles are identification of the foreign organism and at the same time memory. These are the two things that entire immunology revolves around. But basically what we are interested in today's topic is about antigen. Why I have chosen this topic antigen is keeping in mind about vaccination protocols and the methods of recent methods in vaccination, etc. We have to learn what actually antigen is and how many types of antigens are there, and at the same time applications, interactions with the antigen antibodies, and at the same time how vaccination protocols have evolved in the recent days, etc. So let me start first with the first slide. The methods of infection are important whenever you are thinking about an antigen. The methods of infection are usually we get the infections by way of oral pathway and by inoculatory pathway. Second, by injections and needles, etc. And by absorption also. So I will give you some examples pertaining to the first one. Inhalation pathway is by nose. Basically, in the earlier days, they were thinking about only tuberculosis as an organism that enters through inhalation pathway. Now we have several organisms, in the recent one being coronavirus also. Second example pertaining to the absorption pathways are gastrointestinal manifestations, or antigens entering through the portal of intestinal pathway, etc. are called as absorption pathways. Likewise, in case of inoculation pathways, we have AIDS. Usually in good olden days, they used to use only syringes. Therefore, contamination of the syringes resulted in inoculation. And at the same time, blood, etc. whenever there were transfusion, during transfusions also, during inoculation pathways, they used to have this spreading of the infection. That was in 80s and during AIDS time. Likewise, insertion of medical devices. Usually in case of blood transfusions, etc., several devices are available. Now, nowadays, of course, sophistication has increased and plastic things have disposable things have come into existence. But previously, they used to use the same syringes with boiled condition, etc., in boiling water, and they used to use the same syringes. So insertion of medical devices means using the same instrument again and again resulted in spreading of some of the infections, and that was also pertaining to the AIDS. So portals of entry are very important in terms of antigen entry into the body. So let's now examine how many types of antigens are there. First type of antigens, most important ones are protein antigens. Why protein antigen? Because most of the infectious organisms are covered by means of protein overcoat. Some of them with polysaccharides, some of them with lipids, some of them with nucleic acids, etc. But topmost among these antigens, types of antigens, protein antigens are more prominent because protein component and an amino acid sequence, etc., on the topography of the foreign organism is sequestered. So it has some kind of arrangement for the protein, and every organism has its own topography. And that's important. It plays a key role in the detection mechanism by means of our body. And I can give you several examples pertaining to the protein antigens. We can take one example as HBsAg, hepatitis B surface antigen. Only a part of the antigen, part of the protein becomes an antigen, not the total organism. Therefore, protein is important. Protein component of an antigen is important because this acts as an antigenic component. The entire overcoat of the body may not, in case of hepatitis B surface antigen, only a part of the protein itself is a sequence of amino acids itself is important for activation. Similarly, in case of second examples, polysaccharide antigens, we can give the examples of influenza virus, etc. Common influenza virus is a polysaccharide coated organism. Likewise, we have several organisms, but we need not go into all the types of chemicals that are involved in, because sometimes protein and polysaccharide get merged together, or some amino acid sequences are encountered by means of a particular sequence. And likewise, polysaccharide component will be mixed in it. Like that, antigens go on varying, but chemical nature is basically a protein, a polysaccharide, a lipid, a nucleic acid, etc. Next one is, we can classify these antigens depending upon the recognition by our body. First one being T-cell dependent antigen. All the proteinaceous antigens depend on our T cells for identification. Why? This is very important. It needs an explanation for it. Because thymus is an organ that has evolved in the course of evolution in self and non-self recognition. So what is self and non-self is the recognition responsibility by means of T cells. And this T cell alone can identify a protein antigen. T cell dependent antigens means antigens that are made up of proteins alone are dependent on T cells. T cell is a thymus modulated cell, thymus dependent cell, or T cell is called as a thymus dependent cell. This thymus has evolved in the course of evolution, and this has attained a size of about 70 grams in case of adult human beings. Because in young children, it will be up to 15 grams, and as the age increases and they attain the vertical growth and attain about intermediate stage level, and finally they acquire adulthood, etc., it increases to 45 grams. And by the time he attains a maturity and 30-40 years, etc., the organ becomes 70 grams. And it continues up to 70 years of age, or 65 to 70 years of age. And after 70 years, again, this thymus gets reduced. That is called the involution. Why this involution happens is, it is the age-dependent organ. This thymus itself is important in self and non-self recognition, number one. Second thing is, thymus size is also important. In up to 9 to 10 years, this thymus is not that much grown up. Therefore, identification of the foreign organism by means of a child is difficult. But once he grows on, vertical growth is completed, like intermediate stage or in the course of time to about 6 feet, 5.5 feet, etc., thymus gets fully perfect. This is location of the thymus is also important. It is located above the heart, adjacent to the heart. That's why whenever cardiac surgeries or other heart surgeries are carried on, they take more care about this thymus. Because if thymus is disturbed, then automatically it affects the entire recognition mechanism. So our body, that basically self and non-self recognition mechanism is embedded in the thymus, and this thymus has evolved in the course of time, and that has attained higher size in case of human beings, because studies have been made only human beings, not in earlier fauna like fishes or amphibians, etc. Therefore, even though thymus has been reported to be in other organisms, but full studies like T cell classification, T cell receptors, etc., have been studied thoroughly in case of human beings alone. Then next category is T cell independent antigens. All the polysaccharide antigens are recognized by, they are independent. Now, independent here in the sense, whenever T cells are not involved in the recognition mechanism, it is the B cell job to recognize. So B cells always are in circulation. T cells are in circulation, but at the same time, T cells proliferate whenever a protein antigen enters into the body, and B cell proliferation occurs whenever polysaccharide antigens or organisms like influenza virus and etc. enter into the body, then B cell activation occurs. So this should be clear. What is an antigen? What is a T cell dependent antigen? And what is T cell independent? Independent means T is not involved in the recognition mechanism. Directly B cells are taken care of. We'll explain later on how it happens, how B cell is involved in the recognition or in the capturing of the polysaccharide organism, etc. We'll think over at a later slide. Next one is, we'll take some examples of viral antigens, bacterial antigens, protozoan antigens, helminth antigens, etc. Because practically, these viral organisms have become more important because of the coronavirus. But previously also in the initial stages, several viral organisms were there. They were given vaccination and they were given treatment and all that. But perfection is not a endpoint is not there for perfection, because viral organisms also undergo mutation, changes, new viral organisms evolve. Therefore, viral organisms, we can give an easy common examples like poliomyelitis virus. This poliomyelitis virus is an oral pathway entry into the body because of the stool contamination and infection entering into the body, that has been ruled out because of the thorough vaccination as far as viral antigens are concerned. Then let us take the bacterial antigens. Whenever we are talking about bacterial antigens, the best way to remember, an easy example to remember is pulmonary infestation or tuberculosis organism. Tuberculin organisms are best examples for bacterial antigens. Similarly, what is a protozoan antigen? You take malaria as an example for protozoan antigen. Likewise, for helminth antigens, we have filaria as an example for helminth antigens. Because examples we can quote, because basic difference, we must have an antigen, a viral antigen. Poliomyelitis virus is the best example to remember, because the question is remembrance, understanding the concept, and at the same time, why vaccination protocols have evolved in the course of time only to some of the organisms or some of these antigenic stimulants. Why not for all the organs that are possible in the world? There have been constraints in the process of vaccination protocols. So next slide shows what are antigens that are there existing in the body and what are the antigens that are coming out from outside the body. We classify based on that as exogenous antigens. Exogenous antigens are those that are coming from outside, always are called as exogenous antigens, because whatever the viral organisms we have quoted, whatever the bacterial organisms we have quoted, whichever protozoan organisms which I have quoted, and at the same time we have quoted helminth infections, etc. They are all exogenous because they are not inside our body, but at the same time they are coming from outside. Therefore, we classify them as exogenous antigens. The second example is endogenous antigens. Endogenous antigens means the cells have antigenic topography or a red cell membrane, also having an antigen. That's why we are classified as A group, B group, AB group, etc. They are all blood antigens, otherwise called as endogenic antigens, because they are already existing. By the time we are born, our erythrocyte membrane is coated by means of an antigen. It may be foreign to other individuals, but as far as our existence is concerned, we are born with A group, means a particular type of antigen is coated around the erythrocyte membrane. Therefore, they are called as the blood antigens, or they are endogenous. Endogenous means already we have inside our body, not coming from outside. They are, because by the time we are born, our blood group is retired, our erythrocytes are formed. Therefore, they are already existing antigen on the surface of the blood cell. Second kind of endogenous antigens are whenever an organism, organ transplant antigens also are called as, there is a classification here. First, let us examine the blood antigens and then go into the organ transplant antigens later on. Because later on, whenever we are talking of transplantation antigens, we can think about Now let us consider the another category of antigens, that is tumor antigens. Usually, this is also a manifestation of endogenous antigens only. Usually, whenever there is a disturbance in DNA sequence or some kind of tumorogenesis, it may be a malignant tumor, because malignancy is the most common phenomena for formation of cancer or tumor formation, etc. This tumorogenesis occurs because of several factors. It may be mutation, etc. During this time, what happens? These antigens are sequestered around the cell membrane, which is called as a tumor formation. So whether this is an endogenous or exogenous, it cannot, cause may be from outside. That is, some cancer causing material must have entered into the body. As a result, it has become a change has occurred on the topography of the cell. Therefore, it has become a tumor or tumorogenesis. Or it may be because of an infection also, a tumor may occur like cervical cancer. So cervix cancer. So like that, we have several examples pertaining to tumor antigens. So we have to classify these antigens depending upon various options. If it is an endogenous one, existing on the cell membrane, RBC. If it is a tumor antigen that is occurring on the surface of the cell membrane, but it is harmful. Tumor antigens are always harmful. Sometimes they escape from the detection mechanisms, tumor formation, the malignancy increases, it goes on destroying the body, and later on it is detected and all that. So that is a different story. But we have to have classification as what is a normal cell, normal antigens, then tumor antigens or tumorogenic factors are there. This is usually a malignant cell. This is the difference between a normal cell and a tumor cell. So this is how we classify antigens based on exogenous pathway, exogenous antigens, endogenous antigens, then also normal cell, that is erythrocytic membrane antigens, likewise tumorogenic antigens, etc. The next one is examples pertaining to the exceptions pertaining to the antigenicity. What is an antigen? Which surface area of the body is not showing any antigenic response. For this also we have examples like lens proteins. For example, let us consider one example as a lens protein. Whenever cornea is transplanted from one individual to the other, it doesn't have any implications because lens proteins are exempted from antigenic issues. It is not considered somebody's cornea, it's transplanted into somebody else, corneal transplantation. Then subsequently, the second phenomena for exceptions to the antigenic stimulation into the other body is sperms. Whenever sperms are entering from one individual to the other, that is from male to the female, there is no response pertaining to the female because uterine area and other particular parts where the sperms are passing and fertilizing, etc., they're exempted from immunological reactions. That's why we have quoted that first we have said antigen, how many types of antigens are there, and what is the type of classification of antigens. We said T cell dependent and T independent antigens. Later on, we have said exogenous antigens, and later on, we have also said as endogenous antigens, etc. Then we have said membrane antigens like erythrocyte membrane antigens. Then we have also said about the tumorogenic antigens, etc. So pertaining to all these phenomena, there are some exceptions. What are these exceptions? If cornea is transplanted, there is no problem because it is an exempted from immunological reactions. It is a avascular area. Because a lens is concerned, it is an avascular one. That's why, because blood is not there in it, there is no response. It is purely a technical aspect as far as the lens is concerned, a technical structure. Therefore, lens is transplanted from one individual to the other. Therefore, usually eye donations are encouraged, keeping in mind that the lens can be stored in the hospitals and they can be given to individuals who are suffering some visual problems. Likewise, as sperms are concerned, when sperms are entering from the male to the female, it doesn't show any kind of immunological reactions because it's an exception in the nature has given. Otherwise, there should not have been any fertilization mechanisms and all that. Therefore, nature has given a benefit that sperms they are not going to have any kind of immunological reactions inside the body. Now, let us come to how an antigen is processed, because we said antigens are different types. These antigens enter into the body by oral pathway, by nasal pathway, by there is secretory pathway. We usually call them as secretory pathways as far as antigenic antibody secretions are concerned. Let us consider some examples pertaining to the antigens entering into oral pathway, how it is taken care of, or buccal area, intestinal area, etc. Usually, we have our body mechanisms to take care of by means of secretory antibodies. So, antigens that are entering through the secretions are taken care of by means of only secretory antibodies. Now, what are these secretory antibodies which respond to the antigens? For example, we take something through the oral pathway and automatically an antibody should be generated against it. Now, all these are taken care of by means of B cells, because secretory pathway stimulation is alone taken care of by means of B cells, and they produce only IgA titer. So our body, if you look at the body composition, on the surface area of the body, second is secretory area of the body, third is blood. So the immunological responses are categorized into the areas. Number one being all the secretions. First, we take the example of intestinal secretions because we eat food and several organisms may be harmful and useful also. Useful are, we are not bothered about useful, like we eat curd and we get useful bacteria into our body, fermentation bacteria, etc. They are useful. But whenever an organism pertaining which is not useful or harmful to our body is entering into our digestive tract, there is a secretory antibody called as IgA titer, and this IgA titer is in huge quantity in the intestinal area. In the secretory antibodies pathway, we have several secretions. First is the intestinal secretion. Intestinal secretion responds quickly, and at the same time, it tries to generate an IgA sufficient enough to encounter the intestinal infections. Similarly, we have secretions coming into the eyes. Now, whenever an organism or microorganism or antigen enters into the eye, then we have secretions coming by way of eyelids, and at the same time, these tears are helpful in preventing the infections that are entering through the pathway of eyelids. We also have areas, for example, cervix area. Whenever the vaginal secretions are there, the vaginal area is loaded with. Nasal secretions are there, nasal area is loaded with IgA. Like that, this IgA titer, it is being a dimeric antibody. We have to examine the antibodies in terms of the structure. There are dimeric antibodies. All the secretory antibodies are dimeric antibodies. They have an enzymatic stimulation whenever it is tagged to the antigen, and subsequently it lyses it. Therefore, it is called as a dimeric one, which is about with like 2G bonding. That is, one Ig is a Y-shaped structure and an IgA is double Ig, reverse YG structure, reverse Y. Two Y's are there in the structure. Like that, we have secretory pathways or secretions are seen in our body in different areas. I quoted only some of them, like nasal secretions. Our infections enter with the nasal pathway. Several infectious organisms like cold, etc. they enter. There are several viral organisms that enter by a nasal pathway also. Whenever these organisms are entering by a nasal pathway, then nasal secretions are discharged. So always, whenever a microorganism or an antigen is entering through the nasal pathway, nasal secretions are released, and then antigen is encountered by IgA related to that organism. The bonding, etc., we'll discuss later on how this antigen-antibody bonding occurs, how it is precipitated, etc. We'll see at a later stage. Then we have cervix area. Usually in the vaginal area, females have vaginal secretions, and these vaginal secretions always are useful in preventing the infections coming by way of during fertilization mechanisms, etc. Whenever coitus is occurring, there is a possibility of infections entering or contamination by means of napkins, etc. Therefore, the vaginal secretions are useful in controlling them. Therefore, secretory path IgA secretions also are loaded in the area of vagina. So we have salivary secretions in the oral pathway. We have nasal secretions in the nasal area. We have in the eye also we have a tears pathway. We have gut also, intestinal pathway. So these are all the examples of secretory antibody productions to prevent the body from entry of the infections. Then we will see examples pertaining to the surface of the body. In 2011, a Nobel Prize was awarded to a scientist called R. Steinman for describing the pathway of detection through the surface. Whenever microorganisms enter through the surface of the body, like bites of the insects or injuries, etc., it is the job of the dendritic cells, which are similar to neurological cells. The dendritic cells are similar to neurological cells. Therefore, they're dendritic in appearance, but they are responsible for immunological reactions also. They recognize, this slide shows surface antigen detection by dendritic cells. The Nobel Prize was awarded in 2011. Now, what actually happens to the surface antigen detection? This was reported in a Nobel Prize was awarded in 2011, but previously it was done. The research was carried out maybe in 1980s, 1990s, etc. And this Nobel Prize was given to 3 scientists. One of them was Steinman, who described the actual pathway. Whenever the infections are prevalent on the surface of the body, they are detected by means of dendritic cells, which is shown on the screen. These dendritic cells in turn communicate to the T cells so that they trigger the response at the blood level and pump it into the surface area. For example, an injury occurs on the surface of the body. Whenever an injury occurs, the macrophages rush in there, or monocytes get transformed into macrophages and reach to the particular point and either engulf the material or secrete or create an environment which is called as aseptic condition by accumulation of several other cells. So macrophage attractant factor, normally called the monocytes which are in circulation, they increase in size and become a macrophage, and this macrophage engulfs the organism or neutralizes the ability of the foreign organism. So the surface antigen detection by dendritic cells also plays a key role, because dendritic cells are immunological cells or immune system related cells, as reported in the latest scripts. These cells detect the organisms like surface infections. Surface infections may be a fungal infection or any other organism, microbial or even surface areas whenever some bacteria are also invade, because minor injuries also result in bacterial invasion. So these have to be detected and communicated to the T cells, because on the surface area, there is no T cell circulation. It is only in the blood that they are there. But how to detect the environment which is there on the surface of the body? Then it was proved that dendritic cells are evenly distributed like nerve cells, and they are responsible for communicating with the infectious organisms to the T cells, so that the T cells will be able to generate an antibody, or T cells will be able to respond by T4 subset synthesis, that is thymus modulated cell corresponding to the foreign organism. This is very important, because whenever a dendritic cell is communicating the T cells, they become, first helper cell is an identification cell to the foreign organism. Dendritic cell is communicating with the T cell. T cell in turn will communicate to the T cytotoxic cell. This T cytotoxic cell will generate a chemical substance, an enzyme corresponding to neutralization of the foreign organism. That's how the synchronization occurs between dendritic cells and thymus modulated cells in order to prevent surface infections. So we have antigens coming by oral pathway or secretory and antibody pathway. Then we have surface antigen detection also. Then the third option is, next is how it happens in case of blood. So this slide is showing antigen encountered by IgA secretions. This is what I told you, that in case of intestinal infections, whenever you are getting some infection, IgA antibody, which is shown as a dimeric one, is focused on the slide. This is the second most common serum Ig, because the highest one being IgG, that is the highest concentration is IgG, because the smallest antibody. The second is the IgA, the second most common serum IgG, and it is a major cause of a class of secretions in tears, saliva, colostrum, mucus, etc. Even in colostrum also, I forgot to tell about colostrum. Usually when a mother is feeding the baby, in the initial stages, the baby doesn't have any immunological competence, because neither the thymus is developed nor bone marrow is developed. So in the infantile in the condition itself, in the 4 months, 5 months, like that, up to 1 year, 2 years, 3 years, etc., till the milk is given, because this milking period varies in animal to animal. In case of human beings, usually they used to give milk up to 2 years in the good olden days. Now it has come down to the level of 6 months or even less also, because now there are several issues for giving milk to the babies by means of mothers. But actually in the nature, what has happened is, the colostrum also has a lot of IgA titer, and whenever milking is done by means of mother, automatically the IgA secretions go into the digestive tract of the young one, try to take care of the health of the young one. Usually you can see, buffalo is milking the young one, and the young one just after the delivery, immediately it goes on playing and in and around the mothers. In spite of exposure to several infectious organisms, The young ones are not having any health issues because of the milk containing the colostrum or the milk containing high IgA titer. Therefore, the secretory antibody is able to take care of in initial stages, that is when the child is just born until the child grows, till her own thymus is formed or till her own bone marrow is formed, because bone marrow formation occurs in the course of age and it increases to once the vertical height is attained maturity, usually 10th to intermediate level, the bone marrow sufficiently increases, or bone size increases, the vertical growth stops. By the time bone marrow attains sufficient size, then it produces all kinds of B cells, etc. Till that time, the young ones are taken care of by means of mother by way of colostrum or the milk. Now let us see the next slide. Third is in case of blood infectious organisms, antigen circulation encountered by IgG or IgM in case of blood infectious organisms. Usually in the blood circulation, whenever a mosquito bites, let us take some examples because that gives you a confidence or strength in order to explain to a student as to how antigens enter directly into the circulation and how it is taken care of by means of IgG. If the proteinaceous antigen is very small, IgG can encounter it because usually free antigens, now whether it is proteinaceous or non-proteinaceous, let us keep it aside because we said T cells are important and recognition of antigens pertaining to the protein nature. Now let us keep that phenomena aside. Now antibody like IgG, how it will take care of minor antigens, small size antigens are immediately captured by means of IgGs. How that happens is IgG will be running in the blood with 80% constant. That is, in the blood, the component is the highest titer is IgG, the major immunoglobulin. Therefore, because its concentration being high, it tries to directly bind to the antigen. And this antigen antibody climax is reached within no time. For simple antigens, I can give you several examples pertaining to the simple antigens later on, because antigens already we have said there are several proteinaceous antigens, non-proteinaceous antigens, lipid antigens, etc. But whenever minute size, here the question of antigenic size, because antigen size means poliomyelitis virus is very small in size. If you compare the bacterial size, bacteria is much bigger in size like tuberculin bacteria. Likewise, you can take tetanus. So like that, several various examples can be explained to the student in order to say what actually antigen is, what is the portal of entry, how it can be taken care of. So we have seen in secretions, a particular type of antibody is able to encounter the antigen. Then in the surface area of the body by dendritic cell pathway triggering T cells and simultaneously T cytotoxic cells being rushed to or being allowed to take care of and pumped into the circulation, that is mature T cells. Third one is in the circulation itself, when something enters into the blood, it is the IgG that takes care of. If the surface area of the IgG is very small for the antigen, then it is the IgM because it is a pentavalent antibody, much bigger in size. Whenever a bigger antigen enters into the body, it is the IgM that captures it, then cells into the cell. Then the B cell tries to trigger, take over the response and produce corresponding antibodies. The corresponding may be IgG or IgM. If the antibody is bigger, more IgMs are produced. If the antigen is very small, IgGs are produced. It's the question of maturity of the cell. So all the plasma cells which are in circulation are triggered by several manifestations, several chemical reactions. But antigen in circulation is always encountered by means of IgG and IgM. Smaller the antigen, IgG will try to encounter. Greater the size or bigger the size, IgM will encounter because the size of the IgM and the quantity also of IgM is also less. IgG is in large proportion, 80%, whereas IgM is very low concentration and it will be able to take care of bigger antigens. This is how the entire scenario occurs in our body as far as antigen antibody reactions are concerned. Now, let us take what happens during accumulation of antigen antibody complexes. Usually, precipitation occurs. Usually, antigen antibody climax will be reached, provided the sequence of amino acids around the antigen is reversed by the antibody form. Let me give you an explanation pertaining to it. Antigen has some protein sequence or amino acid sequence on the cytoplasm. if as long as this topography is existent, antigen will show its effect on the body. Then how it should be neutralized or nullified by means of an antibody? The antibody rotates itself or tries to create an environment so that the amino acid sequence that is there, an antigen is neutralized or nullified by rotation. An antibody surface area can rotate and also nullify the antigen. This is called as an antigen antibody climax. Usually, whenever infection enters into the body, it may take for the recognition to happen about 7 days. That's why doctors advise for any viral infection entering into the body, they are advised for rest for about 4 days or 5 days or 6 days, because viral detection, then proliferation of corresponding antibodies, etc., it takes about 7 days to 10 days. That's why doctors advise not to go for any medication. If it is a viral organism, it will automatically subside, or naturally our body will take care of the infection, etc. That's why they suggest these mechanisms. Next one is, what is an antigenic shift? Hitherto we have been talking about what is an antigen, how antigen enters into our body, how antigens are taken care of by means of antibodies in different areas, on the surface, in the secretions, in the circulation, etc., how this antigen-antibody reactions occur. But sometimes what happens is there is a shift in the antigenic surface area. This is usually an organism after entering into our body, it undergoes a major change, as shown on the slide. A major change in a flu virus resulting in change in protein sequence results in antigenic shift. So whatever the antibody that has been formed, this is called as the first antibody or primary antibody, may not be suitable to trigger the changed flu virus, because it has already undergone a change, or antigenic shift has occurred in the organism. So some of the viral organisms are undergoing drastic changes, as seen in the coronavirus structures also. So antigenic shift has become a common problem. Therefore, during vaccination protocols, this is very important. Antigenic, keeping in mind the topography of the primary antibody response, secondary antibody response also. This is normally called as fine-tuned antibody response. I will give an explanation pertaining to it. You have a lock, you have a key. If the key is perfect, it is called as the most suitable antibody. Most suitable is the key is able to open. For example, the lock is having 7 levers, and if you have a key perfected to the lock, then immediately you can open it. That's how an antibody synthesis occurs as a primary antibody, a secondary antibody. Secondary means the antibody that has been formed after the changes that have occurred in the virus topography. So primarily, the primary antibody may not be fine-tuned one. The secondary antibody may be a fine-tuned one, like a perfect key to a lock. Because locks are opened by people with even wires also. How that is possible? If it touches the lever, it gets open. This is the primary antibody effect. But if it's a fine-tuned or a secondary antibody is perfect, means if the key is also 7-levered, the lock is corresponding arrangement has been made on the key, because key makers always, they take the exact figure of the lock or where the key is going to fix with a soap or something, and they try to produce a key suitable to. Suitable here is perfectness. This perfectness is achieved by means of grinding some of the parts and trying to generate a perfect key to the lock. Similarly, in case of an antigen-antibody synthesis to a particular antigen also occurs in different stages. Number 1, primary antibody, secondary antibody, and tertiary antibody. So like changes occur because the first cell that is present in the body is not fine-tuned or perfect B cell, the matured B cell. We have several plasma cells, but fine-tuned B cell means perfect antibody producing capacity. They try to proliferate. Which cell proliferates in large numbers? It is the perfectly created B cell. How it happens? Because inside the DNA component of the B cell, the changes occur after receiving from the signal from the antigen. What exactly the surface area of the antigen is, it is thoroughly read by the DNA sequencing in the nucleus, and corresponding antibody here is fine-tuned antibodies produced by a B cell. This B cell is called as a perfect B cell or a matured B cell. Then the proliferation of B cells occurs so that this antigen will disappear in circulation. That's why it is taking 1 week time. Usually in infections, why time delay is there? Because if it is a adaptive immune response, that is infection entering into the body, our body trying to respond, etc., it is taking a lot of time. Why 1 week, etc., is this recognition process. First of all, B cell should identify, or a plasma cell should identify, receive the signal from the antigen, generate an antibody like a primary antibody, then fine-tune by DNA rearrangement inside the body, because this antibody synthesis occurs by several genes, and this gene is called as a gene recombination. So after receiving this structure of the antigen, there is a gene rearrangement and gene expression by means of this B cell. The plasma cell becoming a B cell itself is a climax. What is a climax is? A climax is a phenomena where a maturity of the plasma cell becomes into a B cell. Next is, what is the foreignness of an antigen? Now antigens are, we have said some of the antigens are already existing in the body. For example, erythrocyte antigens, as I said, A cell, A group blood people have an antigen called a particular type of protein. It is nothing but a protein, and this is A group protein. Now, for example, somebody is trying to give a different blood group, other blood group. What exactly happens? When the blood group enters, other blood enters into the body, automatically antibodies are pre-existing. So we have to keep in mind that our body is already loaded with pre-antibodies also, then antigens also. The antigen is present on the surface of the cell membrane. Antibodies are present in the circulation. So we have pre-existing antibodies. Similarly, we also have antigens, pre-existing antigens in the body. This is pertaining to the endogenous antigens. Exogenous antigens, this is called as a foreignness of antigen. Whenever an exogenous antigen is coming into the body, it is called as a foreignness of antigen. Now let us examine what is a foreignness of antigen. This foreignness of an antigen is important in organ transplantation. What is organ transplantation? If a particular person is suffering from liver disorders, then can we transplant a liver from a healthy individual? Because in case of liver, a part of the liver can be donated by an individual and that can be sutured or transplanted into the body of the recipient. Because a person who having alcoholic these things, etc., the damage of the liver occurs and a part of the liver gets damaged or totally liver gets damaged. In that case, liver transplantation occurs. So what is the phenomena behind this organ transplantation? How antigens are detected by means of the body? Now here, during organ transplantation, we have to keep in mind that every individual is different. How they are different? These antigens are different from an individual to individual. As we have already said, blood group antigens differentiate individuals. Similarly, other body cells also have difference pertaining to individuals. This is normally referred as HLA antigen difference. HLA means human leukocyte antigen difference. For example, A is having a particular liver cell, B is having a liver cell. Now, how they can be matched? What is called as a matching? That is, during organ transplantation, this antigen, foreignness of antigen is detected in order to match an organ from an individual to individual. This matching is done by means of HLA typing. Now, what is HLA typing? You can simply take leukocytes and compare two leukocytes, donor leukocyte and recipient leukocyte, and compare the antigen. If the similarity is more than 70 to 75% or 80%, it can be easily transplanted. Organ transplantation can be done. Best example for organ transplantation is usually in case of Siamese twins. That is, twins, organ transplantation is 100% accepted. Why? Because in case of Siamese twins, they have originated from the same egg. Therefore, there is no change, much change in case of antigen surface area. Therefore, The twins organ transplantation can be easily done from one individual to other in case of the twins. But twins, the population of twins in the nature is very, very low. It is only the persons with different kinds of HLA typings is more. Therefore, whenever organ transplantation is done, this foreignness of antigen is measured by means of HLA typing, and HLA typing is done is an easy way. Human leukocytes, a little amount of blood is taken, and HLA sequestering is done or HLA typing is done, and compatibility of donor and recipient is tested, and the in a comparative way, the percentage of similarity, HLA similarity is 75 to 80% similar, then the organ transplantation is carried on. Otherwise, they have to wait for it. That's why in case of kidney transplantations, usually people wait for months, years to get a donor. Because if you go on searching for a similar type of a donor, usually it is a rare phenomena that we may have 80% matching or 75% matching. And during this transplantation mechanisms, this foreignness of antigen is very, very important. Then let us consider how many types of cells are there in our body which act as antigen presenting cells. Antigen presenting cells. Number one, macrophages act. First, macrophages. Whenever an infection occurs, macrophages come into existence. Whenever infections enter into our body, macrophages come into existence, and they play a key role in trying to lyse and create an environment. Second cells, dendritic cells, which are also acting as antigen presenting cells. Third cells are B cells, which are acting as antigen presenting cells. Usually APCs, these are called as APCs because antigen presenting cells are macrophages, primary responding cells, dendritic cells on the surface area, primarily responding cells. Then the third category is B cells, which are responding in the circulation. That's how we come across the antigen presenting cells has been classified as APCs, macrophage, dendritic, and B cells. Then let us consider what are autoantigens. Usually we have said that tumors are formed inside the body. Now, what is a tumor? Whenever changes have occurred on the cell membrane and simultaneously a tumor has formed, there is a mutation that has occurred. That's why we have called as a tumor antigen. Similarly, other types of antigens that are formed in the body are called as autoantigens. Whenever an antigen is reacting with an antibody and forming an antigen-antibody complex, in some individuals, whenever these antigen-antibody complexes are not cleared by the body. Now, what is clearance? Antigen-antibody complex should be cleared by the body immediately. This either by means of circulation or in this secretions by means of the excretory system, that is gastrointestinal pathway, or by nasal secretions. They should be transmitted out of the body. But in some individuals where there are some circulatory defects are there, these circulatory defects prevent flow of antigen-antibody complexes and accumulation disorders occur. For example, in the knee area, antigen-antibody reactions occur and accumulation occurs in the knee area. These become antigen-antibody complex mediated autoimmune disorder. Why it has been called as the autoimmune one? Because nothing has come from outside. Some of the antigens that have reacted in the body by means of antibodies in the knee area. Why in the knee area? There is a fluid in the knee joints. These liquids which are there in the knee joints result in the accumulation of AG-AB complexes, and these complexes results in the formation of autoantigens. They themselves become an antigen. That is AG-AB complexes themselves become autoantigens, as a result of which autoimmune disorders occur. So these autoimmune disorders are found in different organs. In kidney also we have autoimmune disorder, because whenever the kidney is not functioning properly, and simultaneously antigen antibody components have got accumulated in the kidney, we have kidney problems. Simultaneously, clearance is not there in the synovial fluid area. This is the fluid that is there in the knee joints. That's why they get limb movement difficulties in that is a knee movement, that is arthritis. This is an autoimmune disorder. Arthritis caused by autoimmune disorder, that is an AGAB complex formation in the synovial fluid. So whenever it is occurring in the knee joints, it results in arthritis-related autoimmune disorders. Whenever it occurs in thyroid gland, it results in thyroiditis. Likewise, in different organs also, autoimmune disorders appear. And based on that, we classify autoimmune disorders under different categories. So autoantigens also are a particular type of antigens. And then we have how long does it take a body? How long does it take a body to produce an antibodies? We said it takes usually 6 to 7 days in order to get signaled, that is a plasma cell to become a mature B cell, or a T helper cell to get communication to a T cytotoxic cell in terms of proteinaceous organisms. So these both categories, that is either T cell response to the antigen or a B cell response to the antigen, they take some amount of time and simultaneously to respond. That's why we said about 7 days time. This is pertaining to the adaptive immunity, infections that are coming from outside for which our immune responses have to adapt. Therefore, this is an example of adaptive immunity. But in case of innate immunity, which we get by genetic constitution, because already I've told you that all the body cells have antibody producing genes and their rearrangement occurs in order to fine-tune the antibody, etc. But basically, our innate immune mechanisms are there by our hereditary features. Therefore, whatever the immunity that we have by means of innate immunity, that responds very quickly. That's why whenever a coronavirus has entered into the body, all of them have not responded. Because whoever had innate immunity, they did not suffer much. But those who have delayed immune responses, because either bones are not sufficiently developed, or subsequent bone marrow is not there, or they have bone marrow deficiencies, or they have antibody deficiencies, because nowadays we never undergo a test pertaining to the antibody titer, etc. It's very difficult, costly also. Usually, a healthy individual is defined as an individual that who has got IgA titer sufficiently, IgG titer sufficiently, IgM titer sufficiently, etc. Nowadays, hospitals are able to measure these antibodies and give a report saying that you have deficiencies pertaining to the immunoglobulins, and they are able to give immunoglobulin injections. But that's quite expensive. Immunoglobulin injections and immunoglobulin therapy is also expensive. Recognition is also difficult. So in order to give a complete picture pertaining to an antigen, I gave you several examples pertaining to the viral. I will give you one more example pertaining to how complicated in case of a viral topography is pertaining to the coronavirus. This coronavirus is the most evolved one. As per our convenience, we are saying that's the most evolved one because it has created a lot of damage to the entire world by way of a lot of financial efforts in order to produce vaccines, etc. This has occurred because the complexity of the viral organism was having spike proteins. That is, the surface area has several spikes, and in turn, these spikes had numerous number of protein arrangements. Therefore, usual normal viral topography, reading by normal viral topography is a response, easy response. But this particular coronavirus entered into the lungs and disturbed the lungs, and basically the lung circulation is low because compared to the blood circulation into the body, liver, blood, organs, compared to the circulation, the lung itself is a very porous structure, and the amount of blood that can be pumped in is very less, in the sense that mostly it is an alveolar area, and if the alveoli are disturbed by these spikes or spike proteins, they will cease to function, as a result of which people have suffered by pulmonary manifestations, and it has resulted in death of individuals, etc., by complexity of the organism. and the vaccination protocols or vaccination production, etc., has taken a long time. Because for vaccination protocols, we have to have the entire topography of the foreign organism, what kind of sequence of antigens are there. Because it was a spiked one, several spikes were there, and that's where several spikes, this surface area to be read, and then a vaccine to be produced, etc. It took a long time, almost 1 to 1.5 years. So even though science has evolved, still we are at the level of not able to read perfectly if the organisms are undergoing drastic changes. Why these changes occur? Because we are disturbing the nature. Maybe this virus has been created in the laboratory, we don't know that, but still people predict, scientists have predicted that this virus was fabricated in a particular laboratory. Some research was carried out, as a result of which it has undergone drastic changes and it became uncontrollable for them to control it, and it has spread and into the entire world and damaged the community, human population to largely and affected. Therefore, we have to have basic design of an antigen structure. At the same time, how to synthesize an antibody, how to have our immune responses. We must have a balanced amount of IgA titer in order to have in secretions. And in order to have IgA titer sufficiently in our body, we have to have a healthy diet, healthy food habits. So if you are taking chemical substances which are responsible for damaging the salivary secretions, etc., then automatically this antibody titer gets diminished. Therefore, we should always take care of the salivary secretions or vaginal secretions or whatever the secretions are there in the body, and also contribute for welfare of the child by milking, as do the animals, because animals are taken care of by means of parental care, by way of giving colostrum and milk, etc., the young one, to see that their population proliferates. Similarly, human beings also have to keep these things in mind, even though there are some exceptions in the nature, but we have to have a thorough discussion about the antigen topography in order to create vaccines. These vaccine protocols are not that easy, but at the same time, not that complex also, provided we have a complete thorough knowledge about the antigen. This is what I wanted to convey, what a structure or types of antigens are there, how many applications are there, because antigen application is more important in organ transplantation, and also antigenic, that is vaccination protocols. In order to produce a vaccine, we have to have a sequestered antigen. In order to transplant a organ also, we have to have a thorough knowledge about the antigen topography. This is what I wanted to communicate by way of my lecture, and I welcome the audience or the participants to pose their questions.
Hello. Good afternoon, sir.
Yes.
Sir, the HLAG will work even for the at the time of baby formation and also for the immunity system. In yesterday's class, ma'am has told that at the time of fetus formation, HLAG will play the important role in the immunity system. So it also pertains for the general body also, sir.
Usually we go for HLA typing during transplantation, because what I said is an application. What is an application is whenever you are interested in organ transplantation, we go for HLA typing. It is easy phenomena, because we cannot go take a particular organ, whether this organ and that organ are same or not. We cannot go for you comparing of two kidneys. That's why we take simple HLAs and compare, because human leukocyte antigens, leukocytes can be taken from the blood, simple send communicate for laboratories and compare, and then try to use for organ transplantation. But babies, it's not application. Babies, it's a different story, because HLA typing is done in order to compare. DNA sequencing is different. You are saying about the child and a mother or child father similarity, etc. That is a different story. Here, antigen means We have some antigens on the blood cells. A. My wife may be having some other antigen. That's not concerned because the baby is important for us. What type of blood group the baby has attained? It may be a combinatorial therapy because A and B, they may combine and give depending upon the allelic frequency and all that. We cannot go into the allelic story because thousands of alleles are there and allelic frequency is yet to be studied in depth because gene frequencing itself is a very complicated phenomena, whereas allelic frequency to be done, thousands of alleles are there on the surface of the body. That's why usually major histocompatibility, MHC1 and MHC2, that is all the nucleated cells have MHC composition. It is nothing but HLA. HLA is only leukocytes we are taking for sampling, therefore we are calling it as HLA typing. Otherwise, all the body cells are nucleated and all of them have MHC class 1 antigens. Now, what is this class 1 antigen? It is nothing but a specific phenomena of arrangement of alleles on the cell membrane. My cell membrane is different from X cell membrane, Y cell membrane, etc. Therefore, we are saying MHC class 1 antigens are individual. Allelic frequency, individual.
Okay, sir. Thank you, sir.
Have you any doubts, students? I hope there is no doubt, sir. Sir, thank you, sir. Thank you for giving this wonderful lecture. And this is now time to give vote of thanks. Respected dignitaries, chief guests, and participants. It is my privilege to propose vote of thanks now on behalf of Department of Zoology, Dr. B.R. Ambedkar University. And on my behalf of, I would like to extend my heartfelt vote of thanks to each and everyone to you for attending this webinar. We would like to express our sincere gratitude to our VC sir, Registrar, Academic Director, and Dean Faculty of Science. You always support and encouraging the faculty members to organize webinars and seminars. Our sincere thanks to our chief guest, Professor Raghuramlu sir. Thank you, sir. Your lecture is very wonderful. We learned many things, and I'm very, very thanks for not only accepting for invitation, but also spending immeasurable time and delivering wonderful talk on this topic. Thank you, sir, and thank you all participants. Thank you all.
Thank you, sir.
Thank you very much. Thank you all the participants for your patience.
Thank you very much, sir, for information.